We're going to talk today about a software package that we developed for gene set testing after we've been evaluating some commercial and open source tools. So before I start, let's get into what is gene set testing. So as we know, when we perform RNA-seq experiment or microarray experiments, so we usually end up with a few hundred genes after we perform differential expression analysis, if we are lucky. If you work with probably some cell lines, you might end up with thousands. And as you know, we've been doing this for probably the last 3 decades. So we've got large databases of pathways, signatures, transcription factor targets, processes, biological functions, and diseases. So these databases are growing exponentially every year. So gene set testing methods basically try to mine these databases and find some relevant gene sets. In other words, processes or functions that can describe your experiment. So you hope if you run one of these methods, you will end up with like 5 or 20, 25 if you are patient. So 25 gene sets that can tell you something about your new biology. Now, there have been a lot of work in literature. So there was a recent benchmarking published in BOSC 1 by Taka, and they evaluated these methods. So they compared a lot of methods, and they end up with the conclusion that the performance of these methods depend on different factors. So most of these, the most prominent factors are the number of samples in your experiment, the size of gene sets that you are dealing with, and the experimental design, whether it is a paired design or unpaired design, and the percentage of genes that are differentially expressed. So to confirm this, we run 12 different methods actually from RNA-seq data, and then we generate here a multidimensional scaling plot. If you are familiar with this, it's kind of a PCA plot for the ranking of the individual methods on 200 gene sets. And then as you say, it's very clear that none of these gene sets agree on one ranking. So they kind of tend to be quite different. And if you know that in literature, we have probably more than 30 methods available. Most of them sit in the bioconductor repository if they are bioconductor. So we need a way where we can basically improve the ranking. So we need to trust, we need a tool that we can trust basically to rank our methods. So to do this, we proposed ECCB. ECCB, which is an ensemble approach that takes normalized expression data, RNA sequencing or microarray from the lemma analysis pipeline, if you are familiar with the lemma and HR packages. And then we feed it into 12 different methods at this stage. And then after that, we take the individual rankings and the individual statistics to combine the rankings and improve the ranking of gene sets. After that, the results of the analysis are taken to generate interactive report. So the methods is already published in the Journal of Bioinformatics. It's appeared last year. If you want to read about the algorithm, you can refer to the article. So currently, as I mentioned, we have 12 base methods. We can add more methods. It depends if we find methods that worth of adding. We also developed another package called ECCB data. And this package basically aim to collect a lot of gene set signatures from literature, so mainly from MSigDB, GeneSetDB, and KEGG pathways. Currently, it includes more than 25,000 gene sets. And these two packages, ECCB and ECCB data, they are both in the Bioconductor open source project. Now, ECCB, the advantage of ECCB over other gene set testing methods, it offers comprehensive reporting capability. So when you run ECCB, usually you will end up with a HTML report. And this HTML report, if you are running on multiple experimental conditions, you will see that each experimental condition in one group. And if you use multiple gene set collections, like some from MSigDB or GeneSetDB, then you will be able to see this report. And then we also generate heat maps and pathway maps using the PathView package from Bioconductor. We also generate heat maps and summary bubble plots and bar plots at the experimental level. So you can see, you can get ready-to-use visualizations. You can take them just copy-paste into your publications. And the most important part is we also generate MDS plot where you can see how the base methods perform, actually, and whether you have a consensus or not. And most of the cases, you will not get a consensus. So finally, how you run ECCB. So to run ECCB, it's very straightforward. 4 simple steps. We tried our best to make it very simple because most of our users are non-computer scientists, so they like easy things like in R. So you need to build and index your gene set collections first. So there are a few functions: build index or build custom if you have a custom gene set. And then you configure the analysis settings, and then after that you run the testing and generate the report. So now we are in the process of submitting another paper in the F1000 research. Hopefully it will appear next week, where we explain in detail how you can run this workflow. So finally, I would just want to highlight that gene set testing leverages existing knowledge to reveal underlying biology, and ECCB make this easy for you. So if you have questions or you want to have more discussions, my boss 143 today, this evening. And if you want to come and take stickers, you are welcome as well. Thank you.